Got it. All right. Well, thank you so much for that lovely introduction and for both to Nancy and Bob for hosting us here and giving you guys all the opportunity to beam into the Carter County Museum. So I am currently in the Lambert Hall of the Carter County Museum, one of our Montana's first mounted dinosaur ears standing behind me. And I've got a lot of fun things I'd like to talk to you guys about. So we'll try to keep this to the agreed upon 45 minutes-ish. And I'm hoping to do a little bit of show and tell, partly because I might not be as zippy at computers as I thought I was. So we might just try to be showing things physically more than a well-prepared slide. So bear with me as I show some real fun things up above with the camera here. So what I am going to be loosely sort of following tonight's talk will be, here we go, tonight's journey. What is a fossil treasure trove? I'll kind of give you a background on why I think feathers are amazing. So phenomenal feathers and where to find them. We'll kind of look at some lithic legends from lakes and lagoons. Talk about the art of the avian amber deal a little bit, and then find some unexpected exceptional preservation in excrement and where I'm looking for it. So what is a fossil treasure trove? And I am going to be sort of arguing that the familiar definition of a Lagerstätten, which is and we'll take a look at a couple of Lagerstätten examples here in a second, but Lagerstätten is a word we give to fossil deposits that have really exceptional either preservation, both with soft tissue or lots of fossils altogether. I think there is a lot of fossil treasure troves that maybe don't meet those traditional Lagerstätten definitions, but we'll kind of see how some of those are fuzzy. So one of the things I really like about the word treasure is that it is an incredibly subjective term, right? And the act of treasuring something makes it a treasure. And I think one of North America's most amazing treasures that we have are our feathers that we can find across here. And this is something that has been noticed through cultures throughout time. So some of the most amazing art that comes from Central America, or the Aztecs, are these amazing feather pieces. This is a headdress. This is probably Montezuma II's headdress. Both of these items are actually now currently in Europe. And Europe really did treasure a lot of the feathers that they found as colonial expansion happened out. For a long time, there was a career path was to be a plumier, someone who just specialized in fancy feathers and how to put them on a hat. So a lot, and in some cases, some of these feathers today, especially from some of these endangered birds, which are or extinct ones, pictured occasionally here, those feathers are worth their weight more than other precious metals. Feathers are really interesting when speaking of weight. They are just a phenomenal integumentary structure that does so much in terms of weight reduction and strength with as little weight as possible. And this is achieved through kind of a hierarchical structure, both within physical things. So if we zoom in physically onto a feather, you'll see that that feather vein has barbs and barbules. These are some terms I might use occasionally. I don't want to get too hung up in all these definitions, but at a very microscopic scale, a modern feather has some amazing features to it that these tiny, simple thing at this microscopic scale, these tiny little hooklets on this correct side of a barb allows for this amazing zipper-like action on a feather, which also allows a feather to come apart and then zip back up together. So it's a really amazing dynamic flight structure, but it didn't start off as that. And one of the questions I've always had is, with my research, is when do we get this truly modern bird feather? And one of the earliest truly modern bird feathers that we thought we found, and still kind of think we do, is the first specimen of Archaeopteryx. So before we even had a full skeleton of Archaeopteryx. This single feather came out of the Solnhofen quarry in Germany, and actually, it interestingly was found by a quarry worker. Came into the publication light because that quarry worker knew that there was a doctor in town that could trade healthcare for fossils. So he had developed chronic pneumonia, probably from working in these Solnhofen quarries that were being mined out for lithographic plates at the time, and occasionally you'd find these great fossils. So this is how one of the earliest feathers came to light, and it really does look like a truly modern feather. It's got asymmetrical vein. This is probably a flight feather of Archaeopteryx. Further specimens of Archaeopteryx really show that these were very much flight feathers. And we've always sort of assumed that in 2 dimensions, the way that we see it, those feathers are more or less a modern flight feather, which is interesting because without the feathers, Archaeopteryx really doesn't look too birdy. Without those feathers, there's without that information, this thing probably would have lived for a long time as an interesting theropod rather than our first known bird. We've since found there's been great discoveries in larger Lagerstätten that are more productive, like the Jehol biota coming out of the early Cretaceous, where we're seeing just amazing feathering of birds. And this is a really nice one here. This particular specimen is an enantiornithine. It has this nice, really fancy long feather. And some of these lithographic deposits do give us really good preservation. So on some of these feathers, we can see individual barbules, but maybe not the hooklets on those things. We can see a rachis, but these are also all very flattened, carbonized specimens that we don't see a lot of 3-dimensional preservation with. So for a long time, there's been sort of assumptions that the rachis that is kind of forming this main stem of all these feathers is probably a modern bird rachis. What's interesting is from this deposit, from the Jehol Lagerstätten, we get this really amazing view of the types of birds that are kind of trying to figure out flight. And there is a wide range of bird flight experiments going on during the Cretaceous. And we now know too that it's not just birds that are doing interesting things with their feathers. A lot of animals that are not quite birds are throwing out some really nice feathers and doing interesting things with them, maybe not with flight. This is a feather, this is a wing from Confucius ornis, and it does show a nice wide rachis there, right? So we get some really nice preservation in some of these lithic deposits. Sometimes we get a whole 3-dimensional feather. So these are rachis feathers off of a enantiornithine. And these long, elongated rachis ornamental feathers often have what we see as a medial stripe that runs down the middle of that. Whether or not that is, what is that? It's always been kind of a mystery. I'd say it still kind of is exactly what that is as a bit of a mystery. Is it a pigment band? Is it an actual groove? And some of these 3-dimensional things, it looks like it's an actual groove, but it's a structure that is pretty common through a lot of these feathers. This is all well and good, but it'd be really nice to see if these feathers have any of those, 3-dimensional microscopic details that make feathers so important today. So where do we go to find that? Amber. This is one of the ways that we can find some really nice preservation of feathers. Amber itself is just an amazing gem, naturally formed gem of sorts, right? It's kind of like a bioresin or a bioplastic of sorts. It's resin produced by trees that is fossilized. And that process seems to preserve feathers super well. It's kind of variable in terms of bones. So it seems like a lot of these resins probably have enough acidic content to them that the bone preservation in some of these resins are not super great. There's a lot of interesting work being done taphonomically on that, but the feathers themselves seem to preserve super, super well. How do we get feathers in amber? Well, that's kind of an interesting case in itself. This lovely illustration by Claire Jorgensen sort of shows how could we capture some of these amazing feathers in a tree resin. And it does happen through several mechanisms that we can observe today in the modern realm. So cavity nesting birds, like somebody over here, these are birds that are either boring into that cambium and actually developing resin flows around that. There are the red-cockaded woodpecker in southwestern US does this, where it induces resin flows around the nest of its tree to get these nice sticky resin flows to prevent snake predation. There are even here more locally, we have the red-breasted nuthatch. Nuthatches will go around the forest and gather up resin from fine trees. And then because they don't typically build their own cavity nest, but they're a very small bird, they will line that cavity with resin to make a smaller hole. And it also probably has some antimicrobial properties as well. In some of these cases where you do have cavity nesting birds in a highly resinous forest, early fledging failures might happen. This is maybe why we're seeing a lot of baby birds coming out of Myanmar. Feeding fiascos, this is kind of where you get incidental capture of feathers. These resin flows, again, are really, so the reason why most resin comes out of a tree usually is from insect damage. And resin is a defense response against insects predating against that tree. A lot of these trees actually not just release resin to cover an insect that's trying to bore into that tree, they also will release pheromones that attract other predatory insects to help combat maybe whatever beetle is attacking that tree. So what's interesting is things like sapsuckers kind of take on, they do eat a lot of sap as they drill in all their little resin wells to make get that sap. But what also happens is resin is produced and they bring in insects. And so sapsuckers actually get a lot of their food from the insects that they harvest off of these resins that they have induced to flow. And then you also maybe get your kind of bycatch, whether animals preening something itself in the forest, or if it's captured another animal that has feathers. If you've ever walked around a city and just seen a giant pile of feathers, chances are, if you looked up at the right time, you might have seen a Cooper's hawk or something plucking all the feathers off of this pigeon. Those all fall down below, and if you've got some nice resin patches on below, you can capture those feathers. Now, the cool thing about all this is, if you've got trash on the floor of a forest, you can actually kind of tell that it's forest floor trash if you've got the right, if you can see the right type of springtails or insect biota that is captured alongside those feathers. So getting back to looking at all these really fun little tiny structures here. Oops. Am I going the right way? Feels like I'm going backwards. Yep, that was the wrong direction. Here we go. Sorry, everybody. I think I'm going, looks like I'm going, it feels like I'm going in circles. Sorry. Oh, there we go. That's the right direction. Maybe. Sorry, I just probably gave you guys a headache looking through all this. I guess we're going to look at, trying to find a feather, see these three-dimensional structures in amber that I was kind of talking about. Now, a rachis itself, just the main stem of a feather, is a super interesting structure. And I can go all night on just how interesting that is biomechanically and how that changes on every type of bird and what they're doing with their feathers. But mostly I want to be focusing on is that most rachises have a square cross section to them and spongy infill pith. And even on these fancy long-tailed feathery examples of today, where we have these very similar racket plume looking feathers, ornamental feathers, they all have very interesting cross sections. Some of them are not always square, especially some of these dancing. If you could see some videos of some of these hummingbirds and what they do with their ornamental feathers, it's incredible. But a lot of that is possible because of this cross-sectional geometry of that rachis. And what was interesting, some of my research at USC was looking at some of these feathers from Myanmar. And when we looked at what looks like a pair of ornamental feathers, they've got a nice wide rachis. It looks like there's a medial stripe there. It looks like what we've seen in lithic fossils. So let's do a 3D scan of that. We did a micro CT scan. And it turns out that that didn't have the cross-sectional geometry that you might expect for a modern bird feather. When we looked at a lot of other feathers, also from Myanmar, so about 100 million years ago, we found a lot of examples where there just isn't quite that modern cross-sectional geometry, where you see any semblance of a square cross-section, but also any medullary pith. And some of these feathers really do seem to be incredibly and almost impossibly thin, but it's a common enough structure that this is, I don't think, a taxonomic thing. And I can go into the depths of that for a reason, but what we kind of proposed was that there was a different developmental pathway that was present in the Cretaceous, and possibly one that just wasn't utilizing today's modern medullary pith. There's also so what's really cool about medullary pith, real quick, is that that is what allows these feathers to form as complex and as long and as wide as they are. This medullary pith is really interesting in terms of when it forms. So most feathers kind of are, they're formed or divided somewhere in the dermis here. What's interesting is the medullary pith itself actually doesn't start expanding until it's in that pin feather of a developing bird feather. And it's that expansion of that pith that really forms that cross-sectional shape of the rachis. So these feathers in the Cretaceous don't seem to be utilizing that pathway, which might also explain why we don't quite see as big as bird feathers or maybe as complex of feather shapes in the fossil record. That being said, they're still doing they're clearly flying, and they're still doing quite a bit of interesting things with it. Here's another bird, wing, actually, that has some really amazing feathers preserved. And another CT scan of that kind of shows that the cross-section of these rachises are not as square, again, as we think they would be, right? So fine, maybe ornamental feathers don't need to have that square cross-sectional rachis, but you'd think that the flight feathers of a bird, right, might have that square cross-sectional rachis. And we're not really seeing that like we see in modern bird feathers. So one of our proposals is that medullary pith might not really be present across the board in all birds in the Cretaceous. Now, mind you, there's a lot of different types of birds, and maybe medullary pith doesn't preserve that well, and that's fair. There is not many recorded instances of medullary pith, or none so far, in the Cretaceous, but there are some from lithic deposits in the Cenozoic. So it's not that it doesn't preserve at all. In fact, it does preserve well enough even in our Cenozoic lithic deposits. So getting back to all of that, I just showed you some really amazing pictures of feathers from Myanmar. And one of the problems with amber from Myanmar, and really amber from a lot of places, is that there is an ethical cost to that of some sort, whether that is exploiting local labor or even just environmental costs, right? So this is what an amber mine looks like that is legal, but it's large scale and it has an environmental impact. This is an amber mine in the Baltic region. So these amber in itself has sort of an interesting cost to it. This is probably the most real part of Jurassic Park, was the part where they were blowing things up to get amber out. And maybe not using I can see here that no one's wearing safety hats or respirators. And so one of the questions I kind of had when getting done with my PhD work and having the opportunity to look at so many amazing feathers, both in lithic deposits from China, but all these amazing feathers and things that are coming out of Myanmar and amber, is this really special preservation? Do we have to go through amber mine sources or even just exceptional but rare Lagerstätten to get really good preservation. And one of the hypotheses was that maybe all amber, whether or not it's gem quality or not, has an opportunity to provide information. And this is something that in the Hell Creek Formation, and this is a great figure put together by Stu Cook, who you'll hear about quite a bit. So I'll maybe introduce him in a second. But the Hell Creek Formation is a really interesting spot for its preservation of the KPG boundary, that big extinction event. And it's very interesting for the types of dinosaurs and mammals and things that happen across that boundary. But what's also interesting is that there are some big insect extinctions that happen across that boundary that we don't have good information for, because when we look at where we get insect information from, it's from amber, and we don't have a lot of known amber deposits right around that boundary. Now, when I talk about known amber deposits, these are typically amber that is a polishable grade amber, maybe not gem grade, but for the most part, you could put it in a tumble machine and it would survive. And in a lot of cases, like the Canadian amber, this is amber that's been sort of secondarily reworked along shorelines. So this is amber that has a bit of stability to it. The Hell Creek amber, I'll tell you right away, does not have that. But does that mean it doesn't preserve anything in there? And one of the reasons why even if it's kind of hard to get to, it's an interesting way to get at information, because the Hell Creek Formation does have a large body size bias. This is a great figure from Brown et al. That shows essentially how much do we know about you and how big are you, I guess is a good way to kind of look at this graph. So if you're a very large animal, we have several versions of specimens of these large animals that we have a pretty good proportion of their skeleton. The smaller body size we go, the less and less complete we have a picture of you. So a lot of these animals here are represented by teeth or isolated elements that are distinct to being a dromaeosaur, but it's a finger of a dromaeosaur, right? Versus the multiple tyrannosaur specimens that we have from this region, or the multiple triceratops adult versions of both of those, I might add, sizes. And the Hell Creek Formation is a complicated deposit because it's pretty high energy, and that's why we get this size bias, right? We've got this giant duck-billed dinosaur behind me, and that Hell Creek riverine ecosystem, that's a river that's large enough to bury a duckbill or a T-Rex in entirety, but also large enough to crush a butterfly, right? So we typically don't have a lot of these small animals preserved in great detail. And we also have a little bit of, I guess I want to look at this little graph here as a version of a paleo environment thing, right? So it's not surprising that we have really good information, a lot of turtles and whatnot here. But one of the environments we don't have a great representative of across the board are our coals, or what were the marshlands, or the swampy parts of this ecosystem. Those get preserved as coal seams or carbonaceous shales, and we do get some fossils from there, but especially if it's a really deep, long-lasting swampland, which is going to be your more diverse part of this ecosystem, it's also, so far, hasn't produced a lot of great information in terms of skeletal elements. But that acidic environment is great for amber. So Stu Cook, Stu Cook. There should be like more opera sounds behind this. This is one of our champion volunteers and employees extraordinary. This is a local high school kid, Stu Cook, who volunteered with the museum for many years and is now at the Burpee Museum of Natural History. So these are his images, because he took this project on and did a great survey of the Hell Creek Formation and looked at its amber. What you're seeing here are nice little amber blebs, very, very fragile amber blebs. But some of these, ooh, boy, we're doing the dance again. Things like this, though, even though this is fragile and breaking apart, this is exactly the kind of amber that we want to look at, because this shows that it was running down a tree and was exposed to the surface, which means that bugs had a chance to get into it. He was able to find much larger chunks of amber, so logs of amber. This didn't seem to actually produce as much information, and we can go into in a bit. But we developed a pretty straightforward, and adopting it off of some other researchers too, of embedding amber in an epotech resin, which has a long setting time. So it also has the same refractive index as the resin or amber does. So a large volunteer effort here, was able to embed a lot of resin. A grant through the Red Ants Pants Foundation was also really instrumental in getting us a nice microscope. And what's really great about that is that the Red Ants Pants Foundation was able to fund the discovery of the first ant from the Hell Creek Formation. So this is a finger here for scale, but this is a ant in a piece of amber. We were able to find other things as well. So I talked a lot about feathers, and then I kind of jumped into not feathers here, right? One of the goals here is to find feathers. Unfortunately, all we're finding is bugs. And not unfortunately, because again, we have such a small, there's such a large body fossil gap for insects across the Cretaceous Paleogene boundary that any insects that we find from this area are very helpful. So this is a really fun pupa case. This is probably what that is there. This is a biting midge. This is I forget what kind of bug this is. It's gonna come to me now. I'll apologize, I'm not an entomologist. And this is one of the fun things about this project, is that it really does require a wide group of people to figure out why any one of these things might be a treasure. But this is an aphid of some sort. This is this weird long body part there. That's actually its mouth part. So here's its eyes, its antenna there. And then this is a siphon-like mouth part that it can kind of attach to a plant with and suck out all its tasty, sappy goodness. A lot of the insects we get from this are not all super, super great across the board in terms of having a complete individual specimen, but the preservation is good enough that you're starting to get color variation and color banding preservation here too. So this is the abdomen of an insect. And you can see those different color bands through there. Again, this is amber that is been looked over for the last 100 years of the Hell Creek Formation. This is just, it's really fragile stuff, which is, really, it makes it hard to go from the field to a lab, even across the country with some of this stuff. So the nice thing about having us set up here at the Carter County Museum, so we can get this out of the ground quickly and then embedded before it has a chance to really explode on it. So this is a really nice photo of that ant again. So it's got a nice big head there. There's its antennae. It's missing its butt. So this, we're going to have hopefully to find more of these ants. And in fact, you need to find quite a few types of ants. Because again, at this point, these are colonial insects. So whether this is a worker or a soldier, this is not a soldier, but to really get a good idea of what species of ant is going on there, you need to have multiple versions of these. So we just need to collect more of these. This is the other side of that ant there. And you can see that there is variable preservation. I'm not saying this is the best preservation in the world. And so far, I've showed you some fairly scrappy examples. That being said, these scrappy examples still have, you can kind of see little hairs on the feet there. The preservation here is good enough to really get this is a formicine ant, which do this fun little action. What we find a lot of here are parasitoid wasps and flies. So here's one of those little guys here. Here's his head looking at you. These are potential feather fibers and potential, integumentary structures. Now, I say potential because these are, what I see here is not evidence for a highly derived feather structure. This is another photo there. And this is the tricky thing in the Hell Creek Formation is we do have ancestors to modern birds in the Hell Creek at this time, but we also have several enantiornithine groups, as well as a lot of other, so this is some other research here of other people. This is what I want to talk about. So if you're looking at the fuzzy things that could be happening in the Hell Creek, we've got a lot of fun options here where enantiornithines and ornithorines, these are birds that are capable of flight and should be producing a pretty understandable feather. But we're also going to be running into a whole range of these more quote unquote primitive feather types. And I say primitive because they're maybe not full on bird flight feathers, but at this point in time, things have been feathered for a long time. So if something is adapted for using its feathers for display or for thermal regulation, there's probably going to be some really interesting adaptations at the microscopic scale that we might not be able to recognize right off the top at some of these quote unquote simple looking like structures. So I'm going to jump into where am I going to try to find more of those? And that is going to be in coprolites. So I'm not going to be able to share with you all of the images of some feathers in coprolites yet, because we're kind of trying to get that published. But I can say that the same techniques that we were able to use for amber in terms of using a micro CT scanner to scan a small piece of material are going to are the same techniques that will produce really nice looking feathers from coprolites. So coprolites or fossil feces in general are going to be a really great way to preserve integumentary structures. So I have right here well, probably regurgitate from a hawk. This is a modern one. So I'm going to maybe share screen again and put this guy under the microscope. Let's see here. That's a different thing. We'll show you that later. Oh. Okay, so I probably should have warned people this. So this is you're looking at a hawk regurgitate here. But this is something that we were able just collected out on the prairie, and this is just to kind of show how much hair and bone is in this nice mineral packed tubular structure, right? So this is not a fossil yet, and it wasn't necessarily laid in a spot where it's going to have high preservation potential. Other than it came across my path, so of course it got picked up. But this is a really nice example of the types. So there's a lot of different hair in here, and if I was able to zoom in on that even further, the nice thing about hair, especially mammal hair, is that you can usually speciate out an animal just based off of the scales on the hair. And that's actually been able to be done in coprolites from the Paleocene. So we have a bit of an idea of what multidebriculate hair or some early Paleocene mammal hair looks like by looking at SEM microscope scans of hair preservation in Paleocene coprolites. So I'm very excited now to looking at what's more common than just a crumbly piece of amber is coprolites. So what we find a lot in the fossil record are coprolites. Here's another, this is one I picked up not just this field season. It's kind of a nice ropey one. This is from the Belle-Fourche Formation, which is a marine unit that we have exposed in Carter County. That is, shoot, I'm going to probably misspeak here, but I think it's in the 90 million year range. Fun thing about that is it's pre-mosasaur. So this deposit, which is exposed around Alzada area, this is before mosasaurs exist, so I don't know who produced this coprolite. But the fun thing about from where it's from is I can rule out mosasaur. It's probably a plesiosaur of some sort or a pliosaur potentially. That part I don't care about as much, other than this is a very large animal that would have been able to potentially ingest some fur or hair at that time. Now, in the Cretaceous at that time, who has fur or hair? There's not a lot of mammals out in the Western Interior Seaway. So one way to maybe figure out what the fur of a pterosaur looked like, right, would be to go through and scan a lot of coprolites from the Western Interior Seaway, looking for hair-like structures. Ideally, we'd come across some of those feathers that are probably covering some of the many birds are actually out in the Western Interior Seaway that we don't get good feather preservation from in their skeletal record. So that is, I just showed you a modern bird regurgitate there. This is one from the Hell Creek. And Hell Creek, and I think this is, I will say, I think this is a coprolite. This is a chunk one, so it doesn't have a really good external morphology for me to really make a good, strong argument that this is, right, morphologically a fossil feces. But in cross-section, it does have a lot of the things that we'd like to see for that. So let's see if I can get that zoomed in. It's probably very nauseating for some folks. All right, cool. So just on the edge of this, get that zoomed in a little bit more. There. So this doesn't maybe look like a lot here, but this already has kind of the elements I'd like to see for a good CT scan. So if I'm CT scanning something, this is actually what it's doing is just density differences. So if I have a coprolite that looks like it's got a lot of porosity or holes in it, that's a good sign that that might be a good candidate for CT scanning, because as we CT scanned a lot of these coprolites, what turns out to be, ooh, you can really see how bad my cuticles are. It's not exactly what, by the record, this is not my cuticle now. This is more of the surface of this coprolite, which I will admit right now is not super convincing. Yeah, my show and tell's not going so great, so thank you for bearing with me. Okay, some of these, I had this queued up once. Well, any case, you might have to, I don't want to be the case of you got to believe me, it's there. But there is some features on even a piece of poo like this that make it really a good candidate for having potentially soft tissue preservation in it. So this is a phosphatized coprolite, and the voids within it very likely have some good information about the soft tissues that were hard to digest in this environment. And this is kind of gets into, oh, stop share. No, well, let's move this into this guy. So this is another pile of poop that I'm interested in for another reason. So we're going to kind of jump away from caring about feathers too much for the moment. And this is a nice baculites, actually. So I've got a couple of them here. I don't know if you can see on my screen. This is another one. And it's just a, kind of an infill of a common baculites specimen, right? So this is something, if you're walking along the pure shale, maybe a very common find to find. But what's interesting about a lot of these ones is that it's not just a mud infill. When you look at it underneath the microscope, these are actually infilled with little peloidal structures. And we've got a couple of varieties here. Here's that other one. So this one's full of smaller peloids. And I'm not going to profess to know what is the producer of this. There have been some great publications that look at these, and in one of these instances, they actually have a crab that is captured, that was preserved within the baculites. Again, baculites are a shelled cephalopod. And so when they die, the idea is that these shells fell to the bottom, and that maybe either shrimp or some small worms or crabs are using these as sort of a small little refuge, and gets filled up with peloids. So why is that interesting? Well, it's kind of where we found that's interesting. So these happen to be underneath a really, really big mosasaur. Again, getting back to this is how analog we're going to be here tonight in Ekalaka. Because I couldn't get my quarry map figured out here. But this is a quarry map of a plesiosaur that we have been digging up in the Pier Shale of Fountain County. And this nice skeletal from Scott Hartman kind of shows what we should be having there. And what you can see with all of these jackets is that we've got a nice long string of vertebrae. And actually, we have almost the entirety of this animal, aside from its tail. So this tail is no longer with us. I'm pretty sure this is the only part I know that we're missing. We have a lot of other parts of it, and we've been 3D scanning them as we go along. So I'm going to share a new screen. This will be kind of one of the last little things I show. This is a 3D scan of Vicky the mosasaur, which was found by Heath Caldwell, who had come out for the shindig and was looking for some shells with his dad in the Pier Shale. Found a string of vertebrae, reported it to the museum. We were able to get permits to the BLM to dig this thing up, and we are currently prepping it out. So what you're looking at here is the skull of Vicky the mosasaur. This little region here is its orbit. The sclerotic ring is preserved within there. And then along this big long snout here, we have a bunch of teeth. There's also, I will say, it looks like it's a pretty rough looking fossil. The bone preservation on this is really gorgeous. And all of this sort of shmush you see here, the reason why that has not been prepped off is that I'm fairly positive this has some level of soft tissue preservation there. Either the imprints of scales or I don't know what the preservation is entirely yet. But for that reason, it's kind of we're going low and slow with this and trying to preserve all that we can on the outside of this specimen. This is a really cool one too, because you'll note that its snout is quite a bit higher. Let's see if I can get this oriented a little bit better than the rest of the bones here. So this part here where the lower jaw is, this is really what the bone layer is, and the rest of the skeleton extends out this way. The only thing that does not follow those rules is Vicky's snout. So we actually are looking at this thing upside down, right? So in the field, this is how it was. So I think we have a pretty good case here that Vicky did a pretty hard nosedive into the bottom of the ocean. And you can actually even see that in the way that it cross-sects across. So you can see the original planing of the ocean bed here, and the snout is cutting through it. You can kind of even see the slide lines, the slickened lines of the mud as the snout has been pushed deeper into the mud. Why is that at all pertinent to that discussion I just had with these tiny little peloids in these baculites? Well, these baculites, so what's interesting about this animal is that it wasn't eaten up. As far as we can tell, we don't have a lot of evidence for predation. This thing is very well articulated. Everything seems to be in death position. It doesn't seem like, unless everything but that one tail seems to is intact. So we often see that in an environment where there is an anoxic conditions. What these things tell us, though, is that at one point, right, at least before, at some point before this guy fell into the ocean, we did have an active ocean bottom. And because these things are sitting at the sediment interface, it does kind of give us a little idea of how deep, as we kind of figure out who filled up all these little baculites, and also occasionally we are finding crustaceans in these multiple. So here's another thing to point out here, I guess. These things right here are large concretions. So these are a bunch of concretions that are largely just below the specimen, although in some cases they seem to be maybe kind of interfingering with this mosasaur. This all kind of comes back to it's telling the story of this large, big mosasaur is really only possible if we have all of the context cues. How deep was Vicky when she fell into the ocean bottom there? Was it an oxygenated bottom? How old of time are we looking at here? That is told also by the isolated baculites specimens that we're able to pick up to figure out what ammonite zone we're in. So as amazing as a specimen as Vicky is and her skeleton, it's all of these other little context clues that shin diggers were able to pick up and give to us and donate to the museum that allows us to tell this larger story. So I will stop sharing and go on, I think, long enough here. So thank you. And any questions?
Okay, we're going to stop and
Stop recording. Right.